All right, you're welcome back to part 3 of this topic, transport in animal. The last time we looked at hemoglobin, how this hemoglobin, what hemoglobin is. We talked about their structures. We talked about what we meant by oxyhemoglobin. That's when hemoglobin binds with oxygen. We also discovered that for each hemoglobin, they bind with 4 molecules of oxygen. Okay, so let's look at how these hemoglobin um, transport this oxygen they bind, how they release, and how they transport it around the whole body. Okay, so here we have what we call hemoglobin dissociation curve. Now, when we talk about this curve, now I'm going to interpret this graph for you. Now, look at the statement here. It said, as partial pressure of oxygen increases, percentage saturation of hemoglobin with oxygen increases. Now, what this statement is trying to say is this: the more oxygen, the more availability of oxygen, the increase the chances of a hemoglobin attaching with oxygen. Okay, so we talk about PO2, that is a pressure of oxygen. Now, the more there's oxygen, the higher the rate of saturation of hemoglobin. Okay, to take for instance, when you have more particles within, chances of them hitting themselves is very high, because at that point we say that these particles are of higher concentration. Okay, so that's what this is trying to say. As partial pressure of oxygen increases, so the more there's oxygen within, the percentage saturation of hemoglobin with oxygen will also increase. So the rate at which hemoglobin binds with oxygen will also increase, because there's more oxygen present. Okay, now if you look at this graph, let's look at this graph now. Now this graph is showing you percentage saturation of hemoglobin, and then look at the partial pressure of oxygen. Now here is showing, here is 0, that is beginning. So as the pressure of oxygen increases going towards this direction, as the pressure of oxygen is increasing, that is same how the saturation of this hemoglobin will also increase. Now look at the graph, look at the curve. Now this is S curve, also known as the sigmoid curve. Now this curve is showing you, if you can see at 1, at 2, you can see this is where we are at 2. So at pressure 2, we have 20% concentration. As it increases to 6, you can see if you put a line here, you see, let's assume that this is where it is. So we have about 70 percent saturation of hemoglobin. So as it increases, the higher the concentration or saturation increases. Okay, now here we say at capillary in the lungs, O2 supply is high. So lungs is where O2 is being supplied. So the concentration of O2 within the lungs will be high compared to the respiring tissues. When we talk about respiring tissue, you're talking about the tissue of the body that needed this oxygen. Okay. Now here, say hemoglobin is highly saturated with O2 because concentration of O2 is very high within the lungs. So when blood gets to the lungs, the hemoglobin within the lungs is going to attach to oxygen, and then it will now move it from the lungs again down to the respiring tissue, the tissues of the body where they are being used. Now when it comes to respiring tissue, now O2 demand is high for aerobic respiration, thereby causing the concentration to be low. Now when O2 demand is high, meaning that within the blood there is the O2 within the blood will be diffused into the respiring tissues, thereby reducing the oxygen pressure within the blood. Okay, so that's what it means. So that's what it means here. So O2 demand is high for O2 demand is high for aerobic respiration. So for that reason, oxygen potential, pressure is low, because it is going to be used up. So hemoglobin releases oxygen. Hemoglobin is less saturated with oxygen. That's what it means. Okay, so take note of that. Take note of that. That's what this graph is all about. Within the lungs, oxygen pressure is high because there is more oxygen within the lungs than in the respiring tissue. Okay. All right. Let's look at another factor here. Now, when you look at the graph, like I told you, this is S-shaped graph, which is S stands for sigmoid. Now, the question is, why is it not a straight line graph? Why didn't the line go straight? Why did it curve? Okay, why did it curve? So hemoglobin exhibits cooperative binding, which is also known as allosteric effect. As oxygen binding increases the affinity of hemoglobin. Now, when we talk about affinity, we're looking at how the level of attraction. If you remember, I think chapter 2 or 3, when we talked about Michaelis constant, yeah, we'll talk about the level of affinity when something is designed to attach to something. The let's use enzyme, for example, lock and key model of an enzyme. Now enzymes are specific, so carbohydrate, for example, is specific to amylase. Okay, so when the substrate comes, the enzyme that is fit for that substrate will have high affinity towards that substrate because it is all designed to fit in. Okay, so that's what we call affinity. Affinity is when you have high attraction within those two. Now, when it comes, they said hemoglobin is a big cooperative binding elastic effect as oxygen binding increases the affinity of hemoglobin. Now, remember we said we have 4 molecules of oxygen that binds with one hemoglobin. Now, look at this. Remember, this is still at 0.0. Now, let's look at this. What happens at low oxygen potential? Now, this is within the respiring tissue. Okay, this we need respiring tissue. Now there's low PO2. Now the first oxygen binds with hem group. Now the hem group changes shape. That's what we meant by allosteric effect. Now the word that has to do with shape. Okay, site and shape. Now when the first oxygen binds, now the first oxygen will create we change the shape of this hem group, thereby making it more fit for another oxygen to bind. So you see now at this point the affinity will increase. It is increasing the affinity. Now it might look very difficult. It might be a bit more difficult to bind at this level because there is very low PO2. So in this case now, if you look at this line, if you look at there's what we call steep. The steep here is not that high. The steep here is not that high because there's still 3 more sites that oxygen will bind with. Okay, so the first binding will increase the steep, like to this at this point. Now when it moves on to the second, now at this point, you can see now there's an increasing PO2, that because the first one has binded, so the affinity has increased. Now next one, the binding of the second and the third O2 is not easier than the first one because the affinity has increased. Okay, there is a small increase in PO2. Now this results in large increase in percentage saturation. Now you can see now this is another steep. Now the steep increases. You can see that there is higher steep here because at this point there is an increase in the pressure, oxygen pressure within the hemoglobin. Now, okay. Now coming to this last one at high P. Now at this point is high now because 3 has already binded. Okay, now all hem groups are fully occupied. Now the reason why this place didn't go, the reason why we cannot use just a straight line like this or using a line that the steep is high is because at this point hemoglobin is fully saturated. Because remember, we have just 4 binding sites. And the 4 of them are completely occupied at this point. So that's why we get a plateau. Now this place is the highest level. Okay, meaning that all the sites are fully occupied. That's why we say that hemoglobin is fully saturated. Okay, so this is the reason why we cannot just have a straight line. This is the reason why we have a S-shaped curve. That is a sigmoid curve. Okay, at low PO2 there's a steep. Now as PO2 keeps increasing, the steep will also keep increasing. That means is getting high. But at this point, when it plateaus, when it levels up, that means all the site has already been fully occupied. Okay, I believe you understand that. I believe you understand that. So let's move on. Let's move on. So here we have here is the lungs. So here in the lungs we have um, here at lungs, O2 supply is high. Now binding of O2 is also high. It happens there. Now PO2 is high. That is oxygen pressure is high. Now percentage saturation is also high. So everything is high at this point because this place is the lungs. But when it comes to the respiring tissue, you can see the O2 demand is high for aerobic respiration. Now release of O2, now O2 will be released. That's what brings about the low PO2, because O2 will be released from the blood or from the hem group into the respiring tissues, so thereby reducing the pressure or the concentration or the saturation of O2. Okay, so say PO2 is in lungs are higher, PO2 in respiring tissue are lower. Okay. Now this will create, say, allows more oxygen to bind to be more oxygen to be released. That is dissociation from hemoglobin. This is, say, as blood travel from capillary in lungs to tissue. This is one thing that happens. It allows more oxygen to be released. That is dissociated from hemoglobin. So this is where it's happening. Now, the reason why it happens here is because respiring tissues, they have low concentration of oxygen, and movement is from region of higher concentration to region of lower concentration. So it usually goes from high to low. So because the respiring tissues are low concentrated, it's going to um, absorb oxygen from the blood. Okay, so that's one of the reasons why is that it allows more oxygen to be released, because of the differences in the concentration gradients. Now also affinity of hemoglobin to oxygen decreases. So you can see now, when the affinity decreases, it causes the hemoglobin to release oxygen. Okay, all right. So that's that. So let's move on to what we call the Bohr effect or Bohr shift. Now, this Bohr effect is discovered by a scientist. His name is Bohr. So they gave it his name. Okay, when it comes to this point, they said PO2. Now, the factors that affect affinity of hemoglobin to O2, though. So PO2, that is put oxygen pressure is high. When it's high, it increases affinity of hemoglobin to O2. That's what I said before. When the first one binds, the first one might look a bit difficult, but the second, third, and the fourth will be easier because the affinity has increased. And I've told you what affinity stands for. Then there's also something that happens there when CO2, there's a carbon dioxide. When the pressure of that is also high. So when the pressure of this is high, now this happens within the respiring tissue. This happens within the respiring tissue. Okay. So when the pressure of carbon dioxide is high, it will decrease the affinity of hemoglobin to oxygen. This first one happens in the lungs. The affinity of O2 happens in the lungs, where the hemoglobin attaches or picks up oxygen. But when it comes to respiring tissue, the affinity of hemoglobin to oxygen will reduce. And what causes that is because of the fact that within the plasma there is high level of CO2. So this will result in the Bohr shift. Now look at the Bohr shift. Look at this. There are two lines here. Here we have low PCO2, that is low pressure of carbon dioxide. Now, when the pressure of carbon dioxide is low, this is what you get. But when it's high, it will cause a shift from where it was before to another section, another line. We'll talk about this later. Okay, so that shift is what we call Bohr shift. So what brings about the Bohr shift is the concentration of carbon dioxide. Okay, when carbon dioxide is high, it will cause a shift in the curve, but when it's low, it maintains a certain position. So let's look at this bar shift properly and see how what happens. Now look at the man. His name is Christian Bohr. He does that. He discovered that in 1904. Now the Bohr effects, where the affinity of hemoglobin to oxygen is affected by concentration of carbon dioxide, like I just mentioned. So high carbon dioxide decreases affinity of hemoglobin to oxygen. So where there is more carbon dioxide within, it will decrease the affinity. That it will cause the hemoglobin to lose interest in oxygen because of carbon dioxide. So therefore, high PO2 is needed to meet same saturation, percentage saturation. Okay. Now look at the curve again. Look at the first one. You can see the first one where you have low CO2 concentration. Now look at the lines. Now we decided to use the one at 60. That is when there is a 60% say saturation of hemoglobin with oxygen. Here you have a 60%. Now because there is low carbon dioxide, now when you trace the line, this is where you meet the line met. Now when you take it down, you can see it's on 4. Let's assume it's 4, meaning that where there is low carbon dioxide, partial pressure of oxygen that is, oxygen will be required. The partial pressure will be 4 for it to meet this threshold of 60. For it to meet this threshold of 60, it will need 4 pressure. Okay, but when the level of carbon dioxide is high, when carbon dioxide is high in concentration, it will cause a shift. So this graph is going to shift to this other one. Okay, so now at that same 60, it's going to take about, let's assume, take this to be 5. Let's take this to be 5. Now you can see now when there is low carbon dioxide, the affinity will be high. Now the affinity here, it will require lesser number of oxygen to reach a threshold, because at this point with low carbon dioxide there is going to be high oxygen. With low concentration of carbon dioxide, that means there's going to be high concentration of oxygen. Okay, so it will require lesser amount of oxygen to reach this 60, because oxygen concentration is high. Now, but when oxygen concentration is low, thereby creating a high concentration of carbon dioxide, it will require more. Okay, so when affinity is high, it will require less struggle to achieve to do its work. But when affinity is low, it will require more activity, more energy. Okay, let's take for instance a magnet and a metal. Now the affinity that exists between the magnet and the metal is very high, so that is why it is easy for them to attract. But when it comes to a magnet and a plastic, the affinity is very low, so it will need extra. If at all is going to happen, it's going to need extra work for that to happen. Okay, so that's simply what that is in a nutshell what affinity does here. So here I said for it to reach this 60% threshold, it will require less oxygen, because oxygen concentration is very high. There is much oxygen within. Okay, but when there is much carbon dioxide within, it will require more oxygen. So that meaning that the oxygen level will need to also increase. Okay, so the Bohr effect increases dissociation of oxyhemoglobin in actively respiring tissue. Okay, so within the respiring tissue, like I said, this here is the respiring tissue. So where you have in respiring tissue, we have a high CO2, while in the lungs we have low CO2. So take note of that. So it will require more oxygen for this to happen at high carbon dioxide, while it will require less oxygen for that to happen at low carbon dioxide. Okay, so that's what Bohr effect is all about. All right, so let's move on. Now let's look at how this high CO2 decreases the affinity of hemoglobin to oxygen. Okay, how it decreases the affinity of hemoglobin to O2. Now look at this formula here. Now don't bother about this formula. It's not that difficult. So let's interpret the formula for you to understand. Now you remember we said how does high carbon dioxide concentration decrease the affinity of hemoglobin to oxygen? Now here, this is the carbon dioxide. Now this carbon dioxide is going to react with water. Okay. Now carbon dioxide reacts with water. It will undergo a reaction, a, a reaction with the use of carbonic anhydrase. Okay, to require this reaction here. So this reaction is going to bring about the production of carbonic acid. Okay, now look at the first point. There's a CO2 diffuses from respiring tissue into blood plasma, then into the RBC. Remember, we're looking at how does high CO2 decreases the affinity. Now, CO2 diffuses from the respiring tissue into the blood plasma, and then from the blood plasma, the RBC. Remember, RBC, WBC, platelets, they are all found within the blood plasma. Blood plasma is what holds them, okay? And they are all within the vessel. So when CO2 diffuses from the respiring tissue, it goes into the blood plasma first. Then from the blood plasma, then it will now move into the RBC. Okay. Now, second thing that happens here is the carbonic anhydrase in the cytoplasm of RBC. Remember, at this point now we're in the RBC already. So the carbonic anhydrase in cytoplasm of RBC convert this CO2 to carbonic acid. So it's going to be CO2 plus H2O undergoing this process using this enzyme called carbonic anhydrase. It will convert it to carbonic acid. This is the carbonic acid. Okay. Then after that, and this maintains the steep concentration gradient of diffusion of carbon dioxide from tissue to blood. We talked about that before. Now, after that, say carbonic acid dissociates. Now, this is carbonic acid that comprises of hydrogen, carbon, and oxygen. Now, this carbonic acid dissociates into hydrogen carbonic ion and hydrogen ion. So this is the hydrogen carbonic ion, and this is hydrogen ion. Now, you can see the charges. Hydrogen carbonic ion is negatively charged, while hydrogen ion is positively charged. Now, carbonic acid dissociates into hydrogen carbonic ions and hydrogen ions. Now, this will decrease the pH and is not good. It's not good. This will decreasing the pH is not good. Now, what happens? There's a counterbalance that the body, that the cells put into play in order to balance this up. Okay. Now, look at what happened next. Look at number 4. The hydrogen carbonates will now diffuse from the RBC into blood plasma. Remember, they initially come from RBC, from respiring tissue into the blood plasma. That is the carbon dioxide and carbon dioxide. They will come from the respiring tissue into the blood plasma. Then from the blood plasma, it goes into the blood RBC. Now, then RBC to form carbonic acid, which carbonic acid will now dissociate to form hydrogen carbonate ion and hydrogen ion. Now, this hydrogen carbonate ion will now have to leave the RBC back into the blood plasma. So it leaves the RBC back into the blood plasma. That's why I have this arrow to the blood plasma. Okay. Now, because at this point now, the charges are not balanced anymore because the negative charge is out. So we need to balance it up. So that's why we have the chloride ion coming in. So chloride ion will move into RBC to balance out negative charge. Okay, so this process is what we call the chloride shift. Take note of that chloride shift. So the chloride ion will move into RBC to replace the hydrogen carbonate that left. So it will go there and replace it to balance up the charges. Okay, then after that, the hydrogen ion combines with the hemoglobin. Remember, the hemoglobin is within the red blood cell. So the hydrogen ion that remain will combine with the hemoglobin to form hemoglobinic acid. That's HHB, hemoglobinic acid. Okay. Now, hemoglobin has a higher affinity for hydrogen ion than oxygen. Okay, so because of this hydrogen ion that combines with hemoglobin, is going to create a higher affinity for hydrogen than oxygen. Okay, then when that happens, hydrogen ion lowers affinity of blah blah blah. We talked about that. Now the hemoglobinic acid will also prevent pH from decreasing. Remember, so the pH decreasing is not good, so they will need a way to counter it, to balance it up, and that's why the chloride ion comes into play initially, and then the formation of this hemoglobinic acid will now prevent the pH from decreasing. Now it acts as a buffer. Okay, so the pH is not supposed to decrease, pH is supposed to retain its level. Okay, then the last thing that happens there is that hemoglobin will now release oxygen. Now reason is because the hydrogen affinity, the hemoglobin is now highly affinity, is having high affinity for hydrogen, so it will cause a release of oxygen. Remember, all this thing is just for the oxygen to be released into the blood, as into the respiratory tissue. All this process to get oxygen to be released into the respiratory tissue. So the hemoglobin release oxygen that is carrying from the O2 diffuses from the red blood into the blood plasma and then to the respiratory tissue. So again, let me run through it again. The first thing that happens is that CO2 diffuses from respiratory tissue into the blood plasma. Then from the blood plasma, it goes into the RBC. So when it gets into the RBC, it's going to react with water. It's going to react with water to form carbonic acid. So the carbonic anhydrase is within the cytoplasm. This will convert the CO2 to carbonic acid. Now this carbonic acid now will dissociate to form hydrogen carbonate ions and hydrogen ions. You can see these two results here. So when this carbonic acid dissociates, it forms the hydrogen carbonate ions and hydrogen ions. Okay, now this will decrease the pH, and I told you it's not good. So for that, the cells will need to find a way to balance it up so that it will not cause much harm. Okay, so in the course of doing that, the hydrogen carbonate ion will diffuse out into the red blood cell. Remember, that was what reduced the pH before. So it will diffuse out into the red blood cell, into the from the red blood cell into the blood plasma, and then chloride ion will come in to balance it up, because hydrogen ion is positively charged while hydrogen carbonate ion is negatively charged. So when it leaves, it needs to balance up. It needs to polarize the whole thing again, it needs to balance up. So for that, chlorine ion will come in, will move into RBC to balance out negative charge, which the process is what we call chloride shift. Then after that, the hydrogen ion that remain will combine with the hemoglobin within the cell, within the red blood cell, to form hemoglobinic acid. Now this hemoglobinic acid because hydrogen is what combined combined with it, it will create a high affinity for hydrogen rather than oxygen. So when that happens, oxygen will be released, and then also the pH will also will be normal again. Okay, so when when the hemoglobin is now having a high affinity for hydrogen instead of oxygen, oxygen will now be released into the blood plasma. Then from the blood plasma, it goes into the respiratory tissue cells. Okay, all right, so I believe you understand that. Now this is another structural representation of what happens. What I just explained is what we have here. So this round stuff here is the red blood cell, and we said that CO2 will come in into the plasma, and then it will not diffuse into the red blood cell. So this is CO2 now coming from the respiratory tissue into the blood plasma. Then from the blood plasma, it enters into the red blood cell. Then the red blood cell, it will react with water using this carbonic anhydrase to now produce this stuff here, which we refer to as carbonic acid. Okay, so it will produce carbonic acid. Now this carbonic acid is going to now dissociate to produce hydrogen carbon ions, hydrogen carbonate ions, and hydrogen ions like you have here. Now this hydrogen carbonate ion will decrease the pH within the red blood cell, which is not good. So for the red blood cell to recover from it, they will need to get rid of it by moving it out of the red blood cell into the blood plasma. Now in the course of doing that, there's going to be an imbalance in the charges, so thereby creating a chloride shift. That is whereby chloride ion will be added to replace, to balance up the charges that this hydrogen carbonate ion left with. Then once that happens, the hydrogen will bind with the hydrogen ion will bind with hemoglobin to form this hemoglobinic acid. So this hemoglobinic acid to form the hemoglobinic acid. Now the hemoglobinic acid at this point is now having high affinity for hydrogen. Is having high affinity for hydrogen, and because of that, it will cause a release of oxygen because the affinity of hydrogen is high. So that means that oxygen will be released. Now you can see now these are the 4 molecules of oxygen which will be released into the plasma. From the plasma, it goes into the respiring tissue. Now CO2 is only transported in the form of hydrogen carbonate ions, only 85%. Now the hydrogen carbonate ion we mentioned is just 85% of the whole CO2. That's what it means. 85% of the whole CO2 is what forms the hydrogen carbonate ions. Okay, so what happened to the remaining ones? So let's see the remaining ones. What happened to the remaining ones? Okay, we mentioned 85% is hydrogen carbonate ion. Then look at the 5%. 5% dissolve in blood plasma. Now 5% will dissolve directly in blood plasma. Now this remains there as CO2 molecules within the blood plasma. Then the remaining 10% we form what we call the carbamino hemoglobin. That is from the word carbamino hemoglobin. That is from the word we have the carbon, we have the amine group, and we have the hem group. Okay, so this diffuses into the blood, into the red blood cell directly. Now this binds to the stamina amine group. Stamina means end, the ending part of amine group. We've done that before. We've talked about the amine group, and the amine group, we talked about that in biological molecules. Okay, so amine group of the hemoglobin. Now this will form this amino hemoglobin. Okay, like I said, carbon, amine, and hem. Okay, or carbon dioxide, amine, and hemoglobin. Okay, so that's that. So 85% is the hydrogen carbonate ion that moved into the plasma from the onset. Then the remaining 5% dissolves into the blood plasma directly, and they remain as CO2 there, while the remaining 10% forms the carbon dioxide, amine, hemoglobin. Now that means carbon dioxide binds at the terminal amine of hemoglobin to form carbamino hemoglobin. Okay, so take note of that. Take note of that. All right, let's see the last part now. Now, after all this process, remember at the end of the day, oxygen is released. Remember, all this process is for oxygen to be released. Okay. Now, when oxygen is being released, hydrogen will attach to hydrogen actually attached to the hemoglobin to increase the affinity for hemoglobin. So this whole process we just mentioned now will not go in reverse when it comes to transport oxygen to the lungs. Remember, the other one is to bring oxygen to the respiring tissue. Now carbon dioxide needs to get back to the lungs. So all the whole process we just mentioned in a reverse form. So the HHB will release its hydrogen ion there in the lungs to bind O2. That is this again to form the oxyhemoglobin. Remember, at this point it's carrying hydrogen. When this HHB is hydrogen binding with hemoglobin. Now, hydrogen carbonate ion diffuses back into the red blood cell. Remember, this hydrogen carbonate ion is floating within the plasma. So there, at this point, it will diffuse back into the red blood cell. Remember, I told you everything is in reverse. Before it diffuse from the red blood cell into the blood plasma, now it's coming back again. So it binds with hydrogen and converts back into carbonic acid. Now carbonic acid converts back to CO2 and H2O, and then diffuses into alveoli. So the same process back in a backward form. Now secondly, they dissolve CO2 in blood plasma, diffuses into alveoli. Remember the 5%. The 5% diffuse directly into the blood plasma, so it will diffuse back into alveoli, and then the remaining 10%, see, there is one that form the carbamino hemoglobin, will diffuse into the alveoli. Now that is the carbon dioxide is carrying will detach and diffuse into the alveoli, and then from there it leaves um, the through the alveoli into the your nostril, and you breathe out. So that's the process. That's what happens. You can if you look at the diagram here, this diagram explains all this process. All right, I think that is that for this part. So we'll see you in the next part. So in the next part we'll talk about the structure of the heart. Okay, see you then. Bye.